All right. Okay. So we'll be there answering the question paper. That's the 2019 test one biology PI 110. All right. So we'll be very fast and we'll answer this question. This question paper in just less minutes. All right. So pay attention where possible. Be giving explanation, but we encourage you to watch the videos that answer those videos. So okay. So we can go. So you can even register there. All right. The line is 09700 and 920. All right. So for more of these, you can register on the line. Um, so that's my Airtel, right? Just to access more of these because a lot are locked. All right. Okay. So we continue answering the question paper. So these are where questions and we'll answer them very fast where possible. Okay, so the first question says during the S phase, all right, in an animal cells, DNA replication takes place in the centros, while duplication takes place in the what? So suppose we have, we are being asked to say during S phase, which is the synthesis phase of what? DNA. So DNA replication take place where? And also centros take place where? So suppose you have the, all right. So suppose we have the cell like that, all right? These now are what? Um, these are spindle fibers, right? So these spindle fibers that we have here, what will happen is that they are just flowing, all right? So there was a nucleus which actually disintegrated there, a nucleus which formed what? Which resulted into what? Chromosomes like that, okay? So that nucleus resulted into that, all right? So the DNA before, in fact, it actually the replication occur within what? The nucleus inside. So we'll say the DNA will take place, replication will take place in the nucleus. All right. Then the replication of now centrioles, you can see they are not in the nucleus, but they are just flowing in the what? In the cytoplasm. So we'll say this one will take place in the what? Cytoplasm. All right. So then the other question says the set of the full set of DNA instruction is called what? So it's called the DNA book, or sometimes you can call it as what? Genome. So it's a DNA book or genome. You can call it by using any of those things. All right. Then meiosis in a dash division. Then while mitosis is the dash division. So what are they asking here? All right. So they're asking us what? To actually um um I said let me just be lighting answers there. All right. So for one we said we have what? The DNA will take place, replication will take place in the what? In the nucleus. Then the replication of the centros which we are just throwing in the cytoplasm will take place in the what? Cytoplasm like that. All right. Then question 2. The full set of DNA we said is called DNA book. All right. Or you can the best word you can use is the genome. All right. So that's the best answer there you should use genome or DNA book. Then question 3 state mitosis is a dash division while meiosis is a dash division. So what happens that as a result of mitosis, after we perform mitosis, what will happen is that we'll have equal number of chromosomes, all right, So if a cell is this, then the cell divide, everything will be equal into the daughter cells. If we had two number of chromosomes, even that side would be 2. So this will result into 2n, all right, which is the both, which is just two number of chromosomes. So this division, will call it as what? Equal division. While this meiosis will be there to have the number of chromosomes. Let's say you had 10. In the product, you have what? You have less number of chromosomes, and that would be 5. All right, so it halves the number of chromosomes. So we therefore call it what? A reduction. A reduction what? A reduction division. Okay, hope that is clear. Then the question 4 now it says, um stages between two meiotic or meiotic division is called what? So when we just have, let's say, prophase, all right, we have metaphase there, all right, we have anaphase there, we have telophase there. So these stages here, we call them, they are interrelated or they are in between. So we'll call it as what? Interkinesis is just what? So they are inter. When you see something between, that's inter. So we call it inter what? Uh, Interkinesis. So kinesis is just a process of what? Separation or dividing. That's the meaning of kinesis. All right. So that is it. So this one will be what? Kinesis. Interkinesis, by the way. All right. So it's called interkinesis. Interkinesis. So depends with where we are coming from. All right. I'm even like writing the I'm writing the long things here. Okay. All right. So that's the meaning there. Then question 5. Asexual reproduction from a single parent is based on which type of division? So they're asking which type of division result in the what? Asexual reproduction. This is asexual. So asexual will always bring what? Such as binary fusion. There are also, those are types of what? Asexual reproduction, or um, um, so that is it. Yes, binary fission is one of them that will result in asexual. Like the cell won't even perform any sexual intercourse, but it will just divide like that. All right. So that type of division will be based on mitosis, whereby we are not undergoing what? Undergoing any mating or just sexual intercourse. So for meiosis, it concerns with what? Sexual intercourse, which just are gamete cells. So we are there to say sexual reproduction will be based on the what? On the where this question is tricky. They wanted to know the type of division, so you call it mitosis, or it will be based on the mitosis type of division. All right, hope that is clear. Let's go to the next page. All right. So question 6 states meiosis usually result in the formation of what? So an identical or four unidentical genetically and dash cells. So these cells are what? Are unidentical. So I'll say it will result in the formation of four cells, four, all right, four cells or four daughter cells. Then genetically what? Genetically. Then and what? They are unidentical. So different cells. So say different. They are not same as compared to what? Um Compared to mitosis result. So we say now for daughter. You can say daughter cells. All right. Then for daughter cells and I'll just say which are what? Different. All right. Or unidentical. So you can use all those words. All right. We can say an identical. All right. So all those you can light those things. We are running out of time. An identical. Then we have question 7 there. All right. So question 7 say the proid of somatic cells is what, and the pro and that of gamete cell is what. So somatic cells are body cells. We call them autosome cells. All right. So they are autosomes. Okay. So they are called autosome cells. All right. So the proid of those cells now, since it is out into two daughter cells with the equal number of what chromosomes in the process of mitosis, the somatic cells will undergo mitosis process. Then the gametes will undergo meiosis. All right. So since these guys, the proid now or the result, this one it will be diploid. All right. Like two things will be formed with the equal number of, but this one it will be just the half. So we'll call it a haploid or haploid. Um, all right. So we'll say diploid. Then haploid like that. So that's about it. So question 8 will say, um, um, so we'll talk about the meiosis division. I mean, the video is there. You can watch the video. All right. Then, the other question 8 says, in which cell organelles would RNA polymerase be found? So where will be RNA polymerase be found? You know these RNAs, DNA, they are found in the nucleus. So just go there and find what? Light nucleus or nucleolus, because that's where let's say the nucleus is there. Then inside there is a deep fluid called what? Nucleolus. Then this is just, there are even pores there. All right, they are pores there. So there is also some kind of cytoplasm there. So we'll call that as the nucleolus. All right. So that nucleolus, that's where we find the what? The RNA and also the DNA. So in the so in which the organelle cell would do. So it will be found in the nucleus, so just the nucleolus. So I would deeply specify nucleolus because I know they are trying to say in which will be polymerized. So these polymers, they are detailed things whereby I need to give the details one inside, not nucleus but nucleolus. All right. I'm just imagining of giving that answer, but it can be nucleus or nucleolus. All right. So then question 9 say the endoplasmic reticulum is an extension of which membrane? So if you have a cell there, there's a what? A what? The same nucleus I'm from talking about. Then there will be something like that. All right. So, and that's what we call now a endoplasmic reticulum. So those endoplasmic reticulum, they can be floating or just holding on the nucleus. All right. So they are one of the extension, like they extend from the what? Nucleus. So we'll call them what? Um, um, so the membrane that actually have the nucleus, the membrane that it has is what we call what? Is what we call the nuclear envelope. All right. So since this is a nuclear envelope, so we'll write the answer as it will be an extension of what? Nuclear envelope. All right. So it's an envelope. All right. So that's where we'll find the what? Um, that's where we'll find this guy called um um the endoplasmic reticulum. It will attach itself on the what? On the on the envelope of the nucleus. Then question 10 says, what is the law of the contractile vacuole in protists? So protists are just single-celled organism. So this is a single cell. So these guys are found in water, most water bodies, let's say swimming pools, other streams, everything like that. So those guys, they are just single-celled, and they are under eukaryotes, so they have actually a what you call, it's just more like a vacuole. Okay, what prevent, they are always in water, but what to prevent them from bursting, because cells will gain water when they are put in water. So is what we call now a um a contractile vessel. So they would have bursted, but because of the presence of contractile, so it contract, and also being thick, like we know um the structure of cell, which is made up of some proteins or some fibers that are strong, that are hard, or um they are resistant to what, to uh, to other stuff's damage, everything like that. So contractiles are vacuole. It's a vacuole inside it that actually prevent it from bursting. So it prevent what we call now osmoregulation. So in short, it is in charge of osmoregulation. All right. So osmoregulation. Then question 11 says, which type of microscopes would we use to study the change in the shape of living white blood cells? So as the white blood cells, that process of engulfing something, so that change in which they are making themselves a amoeba movement, how do you call it? I mean, what type of microscope can be used actually to view cells, in short? All right. So for this case, as for me, um I can use the light microscope, all right, because this one will be there to look at, these ones are light. So we'll say the light microscope will be there to what, to view cells. Then the other question now says the details of surface texture of hair. The details, you want to really get the details of what, a surface air on top of the air there. All right, like the way you've touched it. So what type of like microscopes can you use to really see the texture type? So the detailed microscope I can use there is scanning electron microscopes. That's a very powerful, it deals with electrons. Remember, there are electrons in the air. Just try to charge your pen and lift it on your book, it will lift that page of your book. So these are electrons are found. So in short, we are talking about electrons, if you don't see it. So we are saying now scanning electron microscope is the one I can use. So scanning electron microscope is the type of microscope that I can use here. All right, hope that is clear. Let's go to the next page. So suppose now we're given question 12. What structure in eukaryote cells is more easily seen in DC or DIC than in what, bright field microscope? Okay. So differential interference contrast microscope, that's differential interference contrast microscope, than in bright field microscope. So what structure eukaryotes, in eukaryote cell, in those cells, normal cells that we have, multicellular cells, what structure can we see uh, than in the microscope? So this bright one is the one we can use, in fact, to do what, to view some, like, let's say cells, everything like that. So now one something that would be differential interference contrast microscope is one of the best or powerful microscope. So this type of microscope actually will be there to view cells, right? So we can say um we can say nucleus. The nucleus, because these cells, these microscope are in charge of what? Viewing nucleus. I mean cell organelles. So the nucleus will be seen in that. Okay, what organelles serve as primary packaging area for molecules that will be distributed throughout the cell. So it's a primary storage. This organelle is also called what? Golgi bodies or Golgi apparatus. They have what? They have two layers. On top you have like you see disc like that, or just tube or bags. Again down there they are bags. So there is primary and also secondary. So these ones they can store as primary packaging. So once they package as primary, these guys we call them Golgi apparatus or Golgi bodies. All right. Okay. So now then question 14 says the head of glyphosate is made up of what? So one of them is made up of phosphate group. So say phosphate. So this guy is where now um we'll see something like a tail and also a head like that. So a tail now won't will interact with water, but the head it won't. So the head is hydrophobic. Then this one is hydrophilic, the tail. So that's the reason why you're able to see salad or just some oils once you put in water. The oils, that's the head part. So phospholipids are made up of phosphate. So the other thing that they are made up of is the protein called choline or just glycerine. Okay, I mean glycerol molecule. So if I'm not mistaken, so there's phosphate and also choline is a molecule. All right, or just can you just write glycerol molecule? So glycerol molecule, it's actually it's the one which makes up what? Choline or just the glycerol, but put glycerol molecule is the one what that makes up what? Glycerophospholipids. All right. So we have glycerol and also phosphate. All right. You see. So that is it. Then we have 15 there. So the hydrolysis of lipids gives rise. In short, once we actually break lipids, what will be the product? That's the question here. So once we break these lipids, we'll get what we call fat acid. All right. So the hydrolysis of lipids, all right, lipids and let's say lipids plus let's say water, all right, we are trying to hydrolyze. So what to form? In short, we are breaking down. So we form, we are like the product of lipids will have what we call fatty acid and glycerol. All right, glycerol and fat acid. So we can now write the answer as fat acid and glycerol. All right. So that is it. Those are the things that actually, as a product, or that result in the product, or in the breaking down of what, the breaking down of lipids. All right. So 16 will say when amino acid is actually ionized, this cool group or just COHH bond and also the nitrogen or the NH3 changes to what? So what are they going to change? Where are they? What will be formed once they change? In short, once they react. So what will happen is that this hydrogen will react to go into that. So the reaction happen like, all right, then plus N what, H what, 2 like that. So what will happen that this hydrogen will actually go there, all right? Then will remain with the cool like that with a negative charge because it has given a hydrogen. But this one which has just received the hydrogen will be positive like that, all right? So we have seen now that when amino acid ionized, like we ionize this thing, like we break them into ions. So for us to form an ion, we form this and also this. Okay, the cool group and also the um, nitrate group. Okay, so that's about 16, and we say those are the answers. So those are the product when we ionize these two guys. This one will be what? Will be this. All right. Okay, and the nitrate now will get what? Nitrate like that with a positive charge. Here it's a negative charge. All right. So that's what will happen once we ionize the amino acid. So the amino acid are just combination of these two guys. So these are amino acid when you combine them. So you leave them like this. So those are the linkage for amino acid. All right. So question 17 states, question 17 gives what product are formed when ATP is. So ATP is a storage form of what? Bear in mind that ATP is a storage form of what? Is a storage form of energy in what? For the cells. Cells, they need ATP for them to work. Actually, that's their energy. All right, adenophosphate, that's the meaning there. So adenosine triphosphate, something like that. That's the meaning, or you should know the meaning of ATP, the abbreviation. You are a biologist, so you just have to know all these stuff. So whatever you see, try to go and also know that. It's not everything will be explained. Next time they'll bring to say, what does ATP stand for? So you see. So whenever you see a new things in your answering or just in your notes, Google that word, please. Then the question says, what product are formed when ATP is hydrolyzed? In short, we actually work on this ATP. What are we going to form? All right, what are the products? So the products will be, one of them is energy. I said this is the energy for what? For cells. Then the other thing that will happen is we'll have what we call now ADP, which is adenosine um diphosphate, something. This was triphosphate. This is now ADP. All right. Diphosphate, that was triphosphate, something like that. Then apart from that, we'll form some pies, like a pi bond, something like that. So we'll form some pies. All right. So the repeating units in both DNA and RNA are called what? So once we see, they say the DNA moves like this. All right. So the pattern that repeats again, let's say, so the pattern that repeats there in both DNA and RNA is called what? So we are here to actually talk about, we saw how, especially for the case of DNA anyway, or chromosomes formation. We saw like histone reacting with the DNA. So the DNA now will go into what? Nucleosome. Nucleosome, nucleoid. All right? A nucleotide. Nucleotide, chromatin loop. Chromatin loop, then chromosomes. So now we'll actually see that in this one, in both of these guys, the repeating unit is what we call now a nucleotide. So the nucleotide is what we call the repeating unit. Okay. So it's called nucleotides. All right. So there will be nucleotides there. Then in eukaryotes, fat acid breakdown breaking down occurs in what? In the what? So where is in eukaryotes fat acids? All right. Um, so this fat acid, they result in the as what? Mostly in the DNA things. All right. They are acids. They carry genetic information. So what will happen is that I mean in eukaryotes, fat acid breakdown occur in what? Where do they occur? So what you need to know is that these fat acid, they give large amount of what? Energy. So for them to be broken in a cell, like um we have something like a mitochondria there. So mitochondria is the what? The powerhouse. So that's where energy is what? is produced. So meaning that these fast acid are the one in charge of giving what higher energy. So the breaking down should take place in the what, in the powerhouse, which is the mitochondria. All right, so that is about so we are saying in the what, mitochondria. Then name two isomers of glucose. So we are talking about now the monosaccharide of glucose. Glucose is a disaccharide to say. So the isomers now we're showing now the the reactant of what, glucose. So what are the things that will be formed that makes up glucose? So one of them is what we call galactose. So watch out the video on micromolecule, you'll be able to see that. So galactose and the fructose. So these are what, monosaccharide. So they form what we call now a glucose that you're able to see there. So that's about this. So thank you for actually reaching this far. It's not easy to watch the video and also have this opportunity to see this knowledge. So what you do is we release these videos just to give you at least you brief a bit, but we lock them. Okay. So question B now. There are three checkpoints in the cell cycle. Briefly explain the importance of each of these checkpoints in the cell. All right. So the three checkpoint that we have is one, mitotic or M checkpoint. All right. So at this point, the cells will actually stay more than, okay. So M checkpoint, which is mitotic checkpoint. So M checkpoint. So the cell will be checked for the following in the following. So um so sometimes it is also called, I said this is also called mitotic checkpoint. It can be also called the spindle checkpoint. All right. So it's called the spindle checkpoint. Spindles are these ones which are what, find into this. All right. So we'll check this and also check this. So it's called spindle checkpoint. All right, or spindle assembly checkpoint. Like we are just checking the assembling of the what, of these guys at the metaphase. All right. So M checkpoint, which is the spindle checkpoint, sometimes check for the correct attachment of chromosomes to the spindles. So once the chromosomes are there, they'll be attached into what, these um that's the reason why it's called S or spindle checkpoint. So checks what? Checks the attachment, of who? Spindle fibers or spindles, but spindle fibers. All right. So that's about to M checkpoint. It checks about that. Then the other checkpoint that we can say is what we call G1 checkpoint. So G1 checkpoint check for the DNA damage. All right. So check what, check the DNA damage. Is DNA okay? All right. So for replication to occur. Okay. Then the other thing that this guy will do is we also check the signs of the same DNA. All right. So it will check the signs of a DNA, and um, the other thing that it can also do is it will check the what, the replication of the what, of the same DNA, and also the signs, like I said. So also it will check what, the replication of DNA. So it will check the replication of these two guys. So that is about M checkpoint and G1 checkpoint. All right. So That is it. Um, we have these two type of checkpoints, and we have seen that there is M checkpoint, G1 checkpoint. Do we have to talk about G2 checkpoint? All right, so there is also G2 checkpoint where we check the synthesis or just the replication of DNA. Is connected, so is interconnected. So the checkpoint for G1 and also for G2, they are interrelated. Okay, so um um when we brings about checkpoints, in order for something to happen, there must be order. Just like in Zambia, you can't go to a what to see a president without even the dean of student. You can't see him without what getting the permission from the secretary. Okay, so there must be order for something to be done well. There must be order. All right. So I can also check about. Let me mention about what the same G2 checkpoint. Okay, so we have talked about G2, I mean G1. So this G2 checkpoint is actually there to regulate the cell cycle. Okay, it will occur at the end of the G1 checkpoint. So it will also again check the damage of what. So this one is just a contribution of what. So it will check the DNA duplication and also just chromosome what, chromosome integration. All right, has the chromosome integrated, has it grown? All right, okay, then um that's about this checkpoint. So if this doesn't qualify, then the checkpoint what, because after this is the last checkpoint. So once the cell doesn't qualify, it will arrest that cells. Okay, it will pause that cells um, um from proceeding, so that it can what repair at this point, and it will go at a what G2 gap. So that's one way this G2 checkpoint will actually play a role in this study. All right, so it is very important for the things to actually be checked before they proceed to another level. All right, so G1 and G2, they are interrelated a bit. They are interrelated a bit, but not really that much, because um, they both do the same, somehow same, but the G2 will be there to really prevent that this cell hasn't grown. All right. Um, it doesn't grown for it to go on the what, on the next checkpoint. Okay. So the G1 will be after the S phase, which is the synthesis phase. So on the G1, but let me just clarify something there. Most what we need to check is the nutrients. All right, is in terms of nutrients, the cell led, like, is it healthy? Like, we are talking about now growth factors. All right, in fact, this is my checkpoint. Let me just lab them. Okay, so G1 will be there to check about the what, the availability of nutrients. All right, so the DNA replication will occur at G2 checkpoint. All right, so DNA replication. All right, so that is it. So it will check the DNA replication. Then here we'll talk about growth factors such as nutrients will be checked under G1. So the issue of DNA damage you can also check at this point, but and also the cell signs is one thing that will be checked. Growth factor and also cell um um cell what cell signs will be checked under this. All right. So without even wasting our time, shall we go to the next question? All right. So that is it, guys, and we have no. It's a very simple things, but we just have to see what is. Okay, the next question now says, explain the significance or the importance of crossing over. So this crossing over, it will actually helps us. So why is it important to cross these genes, everything like that, chromosomes? Why do we need to have that? Okay, so helps. So this crossing over, like we cross this gene and also bring this, so it will helps us what in plant breeding. All right. So once we cross over, let's say grafting, everything like that, you know, those are types of crossing over. So plant, um, it will help in plant what breeding. All right. So it will help in what plant breeding. All right. Okay. So since um the plant will breed like that, um okay. So help in plant breeding. The other thing that it will help us in what? In mapping chromosomes. So mapping what? So it will help us in marking chromosomes and also helps in the plant breeding. All right. The other way to actually increase the variation, all right, vital for ovulation or in several production. So increase what? Variation. So as we cross over, we will increase the different species. Some species will be mad, just like people. If we have a lot of people across China's and everything, we'll create even people with what huge bodies, tall people, everything like that, because of the crossing over. So it will actually also helps in the establishment of what the concept of what linear arrangement of genes. So once we keep on crossing something, again we cross this, let's say Laws and Tonga, Bemba and Nyanja, white and blacks, everything. We are crossing over genes, so it will help in the arrangement of linear like that. So we will arrange them, so it will help us in the establishment of the concept of linear arrangement. So there is what we call linear arrangement, whereby we have been producing the same thing, we have been producing the same thing, then it will help us what as we cross over, it will help us what to establish this linear arrangement. So that's one point you can write on your own. So it needed 4 marks. So you can also say um helps in the establishment of what linear arrangement. So it will help us in that it to establish that. Okay, so thank you very much. That is for question one. Question two. So question three now reads um we are answering question three, which states a classmate proposed that mitochondria and the chloroplast should be classified in the endomembrane system. Argue against the proposal. So we are against this proposal. So why should we? The proposal says these two guys or two organelles should be classified in the endomembrane system. All right. So now, as for me, I can say, the way I can now, the way I can support this is that uh, chloroplast and endomembrane. So we are now argue, like we are against the what they propose to say they should what they should be classified as what endomembrane. So I'll say no, they are not. So these guys, they are what um. So my answers will be what chloroplast and the mitochondria, I would say they are not derived from this endo. Where is the system? They said what? Endomembrane system. They are not even connected physically via what? The vesicles of this endomembrane system. they are somehow some auto independence. That's the reason why we are discouraging them from this concept. Okay. So not, I'll say, they are not coming from that. So they are not coming from, I'll just say ER. Okay. Then in the endomembrane system. Then the other thing that I'll say is they are not even connected. So I'll say not connected directly, all right, to other systems within the cells. So not connected directly to what? To transport vesicles. So like in terms of, let's say, if it was a tree, then xylem and phloem are not connecting these. Let's say they are just different leaves on a certain plant. So not connected direct to transport system of what? The same endomembrane system. Endomembrane what? System. Then the other argument or the other support that I can get or give as against, I will say they are structure which are different. So they are what? So I'm talking about chloroplast and what? And the endomembrane and the mitochondria to say. They are, right, they are different from, so they are different from the vesicles of endomembrane system. So they are different from the vesicles or the structures of this endomembrane system, guys. All right. So endomembrane system, guys. Then because these other membranes, they are actually bounded by what? Single membrane, but for these ones they are double membrane. So they are double. It's more like they have their own cell wall. So double membrane. These others, they have only single membrane, let's say cell membrane, but these ones, they have also the extent outside the part. It's more like we have a cell, right? I mean a plant cell. Then there are the chloroplast, everything like that. So these ones, they are independent. Like they also have their own structure, um such as the same cell, everything like that. But I just mean that they are double membrane. I'm not saying cell. So that is it. So then question 4, mitochondria and chloroplast are sometimes referred to as cells within cells. However, this does not make them completely autonomous. Give reasons why this is so. So I want to give reasons why it is like this. All right. So what happens that there is what we call endosymbiosis. All right. There's what we call endosymbiosis now. Um This endosymbiosis is state that um, the chloroplast and the mitochondria, they existed before. They were prokaryotes. Now what happens that they actually developed, they were just ungified by what? By these were mitochondria and what? Chloroplast. So they were just ungified by what? Prokaryotes, and they became part of that. You see? So that is about 5 5 million years ago. So that's what happened. All right. So now that they were cells on their own, and that's what the theory says, and the theory is what? Endosymbiosis. So it is stating that they were cells on their own, but they were had to what? To be ungified by the eukaryotes. All right. They were prokaryotes on their own. All right. So now what happened was, we are now here to so they had their own liposomes, right? So they had the what? Their own liposomes. So that's about these guys. They had their own liposomes and also other necessities such as enzymes needed um, for the metabolism, or just the process of digestion. Every process of life, they were happening within them. It was happening within them. So most of these enzymes needed for metabolism, we can find them. So this is the answer I'm giving, by the way. So we can say most of enzymes found in the um, needed for metabolisms, they have these guys, these two. They have, all right, have on what? Enzyme needed. Enzymes needed what? Needed for metabolisms. So meaning that they can perform life on their own. So the other thing is that they have own enzyme and also have own what? Um Ribosomes. So these ribosomes, we know they are in charge of what? Digesting proteins. All right. So um So all these organelles I've given, such as other enzymes and also ribosome, they are just within them. Within these two guys, mitochondria and chloroplast, they have these within them. They are not outside. Even if we find them outside, let's say, suppose this is the structure, right? Then we find them, and let's say this is what? A mitochondria, like that. Then we have ribosomes, let's say, on the nucleus there. We have ribosome there. But again, inside this mitochondria, there are other ribosomes, meaning that it is at least these ribosomes, they are used to carry out to digest proteins. So, and we can see again, we are finding them in what? In the mitochondria, meaning that the mitochondria is somehow independent. All right. And also the chloroplast, there are two of these guys. All right. So meaning that everything will be done, such as synthesis of food will take place there. So synthesis of what? Of proteins take place within them. So it will take place within what? Within them, like that. So that's about the answers. You are trying just to support the endosymbiosis theory. All right. That's how we answer this one. You give all those stuffs. So you can watch the video. If you haven't watched the video on the endosymbiosis, the video is there. All right. Okay. So the other question says, describe the characteristics of two polyunsaturated fatty acid. Two polyunsaturated fatty acid. So unsaturated, but there is what we call saturated fatty acid. So those saturated, they have what? Double bond, like that. So the functional group that they have is what? Is like this. All right. So that's the function group they have. All right. So this is for mono and monosaturated. All right. So for monosaturated, there's no double bond, in short. What differentiate then for unsaturated, for polyunsaturated, which is now this case, they have actually the double bond. They have one or more double bonds. All right. So for this one, they have, let's say, a single bond, but in here we find a like more than one double bond. All right. Let's say there's a double bond there. All right. And also there's a double bond there. And also let's say right here, there's a double bond there. So we have more than one double bond there, or more than this double bond, more than this double bond. So there are more double bond in the what? Unsaturated fatty acid. You can even draw the structure like that. You show them like that. That's the function group. And so you do this. All right. So you are explaining now they have more than one double bond. All right. So they have more than one double bond. But for mono saturated, they have this kind. All right. So the other thing that we can talk about is that these the same thing. These polyunsaturated, they have very low temperature. I mean melting point. So very low. They have both have low temperature, but for this one is very low. All right. So for monosaturated, they have also low temperature, but for polyunsaturated, they are very low temperature. They have very low temperature. All right. They are also liquid at room temperature. So they are liquid at what? Room temperature, like that. Okay. So the other thing that we can see is these guys. This is for mono and saturated. All right. So the other thing that we can see is these guys, they actually also what? Um, they can't rotate the bond, which is double bond like that. Let's say once this one can rotate because it's just one. But for this one, once you rotate this one, let's say is not rotating. All right. That is for unsaturated poly. All right. So they have more than one double bond. So no rotation within them. So you just describe anything you know about two double bonds. Okay. So they don't undergo any rotation because of what? Pi bond. A lot of pi bonds. All right. So, all right. Then that is it. It is the no rotation is because of what? Double bond. We have more than one double bond. So question 6 now, which is the end, is state that describe the two classes of starch, their linkage and also the branching type. So in terms of classes, we have what we call someone even wrote it and also wrote it there. So we have what we call amylose. So in terms of classes and also amylo, amylopectin. All right. So we have amylopectin and also amylose. So in terms of now their linkage, amylose is linked to 1 to 4. I mean 1 to 4. So that's amylose. Then amylopectin. So this is 1 to 4 linkage. Then then amylopectin is one which is branched. So it will have what? 1 to 6 linkage. So the branching type, this one is what? Is not blanched. So in terms of blanching, this one is not blanched. It is a straight. That is for amylose. Then the amylopectin is what? Blanched. So it has in fact large surface area. So I want to explain that. I will just give like I said. So amylopectin, we said amylo what? Pectin, like that. So amylopectin is highly blanched, like it's like this. It goes with oxygen there. So by this, it means that it is providing large surface area. So whatever I'm explaining here, that's the answer. So it is providing large surface area. So once it provides large surface area, so the digestion will be what? Easily, and it will give rise into what? These are starch, by the way. Starch result into what? Glucose. So it will give the rise in what? Glucose. So give rise in what? So give rise in what? Glucose. So the other thing, since glucose is higher now in the body, it will cause even the rise in production of insulin to regulate that sugar. So lies also in the what? In the insulin. You see how I'm explaining? These ones, you drop them. The point now, what I'm saying is just what is correct. So I'm preaching here. So get my preachings. So whatever, I may not lie to what is needed, but what I'm explaining, get as the answer. I'm trying to explain everything. So if you don't get it, then you are a problem. So that's about amylopectin, and for that it will cause more what? Of glucose to be produced, which will result into the rise in the insulin production. All right. With the linkage we said is 1 to 6. All right, linkage meaning we have a lot of them. They are branched like that. All right, we are not having a stretch chain. All right, so therefore, when you eat something, they are easily what? Digested. So in potato, we find about 80% of what? So even whatever I'm saying here is the answer. So even you mention of potato, then in amylose, so let me write amylose there. So amylose. So in amylose, very low what? Surface area. The surface area is not this because it's straight. All right. So straight chain. So in short, not blanched. Then low what? Low surface area for digestion. All right. So provide low surface area for the what? Digestion, and also low insulin production. All right. Then the other thing is the low insulin, low production of what? Um Glucose. All right. Okay, so that's good. Then the other thing that we can talk of is this guy now, since it has very, like the surface area is zero, it will cause all those stuff. It's the opposite of the other side. So the linkage now will be what? 1 to 4 linkage. All right. Surrounded, it's a ring now which will be surrounded by what? Oxygen, like that. We'll talk about the structure, don't worry. So 4 linkage. Then the other stuff that we can say is we find what? 20% of these amylose in potatoes. So that's why it's advisable to eat potatoes. You have energy when you eat potatoes, a lot of potatoes. All right. So once what happens that on this one, so have oxygen there on the opposite poles. All right. But for this structure there, all right, the oxygen will be on the same, on the what? Same side, like that. Okay, so that's about these guys. They are whatever I've just talked, know that is an answer. Otherwise, thank you very much for actually reaching this far. It's not easy. Please subscribe, and also um um contact me like whenever you need help. So that's alpha. So 09700200920. So we do even tuition online. We do a lot of courses online. Thank you very much for actually um subscribe to our YouTube channel so that you can access a lot of them once we post. Okay, bye.